physiology, one of the most important reaction. Third screen window. Okay, is it now? Okay, slide is visible to you.
Yes, sir.
Slide is visible.
Yes, sir.
PPT. Okay. Very good afternoon to all. Today we are going to discuss about the very important topic. That is the main basic reaction, life-giving reaction. Very important life-giving reactions that we are going to discuss. Very fundamental, very important from the 8th class onwards up to hello. What happened? Please unmute. Whenever I ask any question, you please interact with me. Okay?
Okay, sir.
Done. Now we are going to start the main fundamental reaction, basic reaction, which is very useful to all the living organisms. The life-giving reaction. This reaction giving the life to all. The simple organism, that is, the chloroplast. That chloroplast is the factory, bioreactor, bio, biological factory that absorbing the carbon dioxide, you can see, observing the carbon dioxide and intake of the water from the root and through the sunlight and releasing the oxygen. And also, what is the main product? Carbohydrate, glucose.
Carbohydrate.
Oxygen is releasing as a byproduct, and the main product is
Carbohydrate.
carbohydrate. That carbohydrate only is the main source to all the living organisms to eat and to survive and to get the energy. Main source of energy that we are getting from the plants, directly or indirectly, that we are getting from the plants. Yes or not?
Yes, sir.
You know, simultaneously, ultimately, we are getting the energy from the sunlight. The all overall energy source is the sun. From the sunlight, only 2% of the light, how much it is? Only 2 percentage, only 2% of the light are being used by the plants to produce the food material and also as a energy products, and also for our breath, oxygen, for our inhaling oxygen. Inhale all the living organisms that inhale the oxygen and exhale the carbon dioxide. You know, the plant also inhale the oxygen and release the carbon dioxide in respiration. In all the living organisms in the respiration, it inhale oxygen and release the carbon dioxide. Even plants also. No, all the living organisms. Don't confuse. Here, so many people will confuse. In the plant, inhale the oxygen and exhale the carbon dioxide. No. That is in the photosynthesis, specific reaction. That is there in the specific reaction in the photosynthesis, not in the respiration. Not in the respiration. So you don't confuse. You don't confuse and don't make confuse your students and children, youngsters, next generation. Okay. As you all are, most of them are teachers. Okay. Then you can see the specific reaction, that is the photosynthesis. The photosynthesis, what happening in the photosynthesis? It absorbing the sunlight and carbon dioxide from the atmosphere and water from the
Root.
water through the root from the soil. Water from the soil
Through the
and
roots from the soil.
through the roots. It absorbing. Okay, and it's synthesizing carbohydrates. Main basic carbohydrate is the glucose. You know, whenever we are in sick, glucose we used to inject the glucose or glucon D. The glucose is the primary product that release the energy, immediately release the energy for all our metabolic reactions that will be useful. That energy. You know, energy is the ATP. What is the biological energy that is in the form of ATP,
ATP.
adenosine triphosphate.
Like that.
Yes. Thus, phosphorylation reaction. So the phosphorylation reaction, that phosphorylation reaction happen in the light reaction of the photosynthesis. You know, the photosynthesis are divided into two, light dependent and light independent. Light reaction and dark reaction. You know, the light reaction and dark,
Like
dark reaction. Okay, light reaction will occur, always occurs in the light, but dark reaction, not only in the dark light night. Dark reaction also will happen in the day and night. Okay, dark reaction means not in the night. Okay, it is the reaction. It happens without any dependence of the light. Okay,
ఏంటి అది పడిపోతుందన్న
that's why that's why we are calling a independent, light independent reaction. Okay, the first is the light dependent reaction. Second one is the light independent reaction without using of the light. Okay, main light resource is the sunlight. Main production machine is the where the photosynthesis occur. What is the photosynthetic site? Chloroplast. Correct? Yeah, the chloroplast is the photosynthetic site. Photosynthesis happen in the chloroplast. Okay. In the chloroplast, you know the photosynthetic pigments, chlorophyll, and what are there? Carotenoids, phycobilins. These three are there now, photosynthetic pigments. The question asked photosynthetic pigments. Write about the photosynthetic pigments. Photosynthetic pigments are three types, that are chlorophyll, carotenoids, and phycobilins. You know the chlorophyll again, nine types. I already told you. Nine types of the chlorophyll. What are those? Can you say? Anyone?
chlorophyll A, B,
Uh,
and
A, B, C, D, E, F, G, H.
no, sir. That is not.
Chlorophyll A, B, C, D, E. And then bacterial chlorophyll A, bacterial chlorophyll B, chlorophyll chlorophyll 650, chlorophyll chlorophyll 660. These four are there in the bacteria, prokaryotes, but remaining all are there in the eukaryote. A, B, C, D, E are present in the eukaryote. Okay, these are the nine photosynthetic pigments. So many competitive exams ask the question. Nine photosynthetic pigments. Okay, nine chlorophyll pigments. Nine chlorophyll pigments are there. That are A, B, C, D, E. Bacterial chlorophyll A, bacterial chlorophyll B, chlorophyll chlorophyll 650, chlorophyll chlorophyll 660. Like that. How many pigments?
9.
Nine. Okay, nine pigments are there. You see the basic reaction of the photosynthesis, you know very well. Even from the 8th class onwards, we are studying this reaction. Yes or not? From the 8th class, it started in the biology. the basic reaction, fundamental reaction, life-giving reaction under the light with the chlorophyll pigment. It producing the what it is producing? Carbohydrates and releasing the oxygen. Releasing the
Oxygen.
oxygen. Okay, that is the basic fundamental reaction that is there, that is happening. Okay, these two. This is the main product. This is the byproduct. Oxygen. With that byproduct only we could able to survive. Otherwise, if there is no aerobic condition, the anaerobic era, no living organisms are there. Whenever the cyanobacteria evolved, then photosynthesis started, oxygen evaluation started on Earth, and then oxygen, whenever the oxygen evaluated, all the living organisms are started evolution. Okay? This is a basic fundamental reaction for all the living organisms to survive. To survive. Okay, is the basic fundamental reaction for all living organisms. Okay, you know the total reaction. How many? 6 carbon dioxide and 18 ADP, 12 H2O, 12 NADPH plus H plus. Phosphating and releasing the glucose, and 18 ADPs are split into ADP and PI, and 12 NADPH are utilized, and 6 oxygen molecules are released. 6 oxygen
The
molecules are
slide was not changed, sir.
not changed. The,
Yes, sir.
the
The first
reaction
slide
is
is
there.
only there on screen. First slide is only there.
First slide on. Now, could you able to see this?
No, sir. No, sir.
Photosynthesis reaction. No.
No, sir. No, sir.
Yeah, what happened to this? In the show.
Yes. Yes, sir.
Okay.
Now it changed.
Yeah. Okay. Then I have to keep like this only. I actually I put in the presentation more. Okay, no problem. Huh? Okay. Here you can see the basic fundamental reaction. It is already studied in order to mention again and again. You know, here in our photosynthesis chapter, we are going to discuss the four main 4 units. That first one is photosynthetic pigments. Okay, light harvesting complex. Light harvesting complexes which harvest the light, which absorb the light and release the energy. Okay, ATP and NADPH, assimilation energy. That ATP and NADPH energy we call it as assimilation energy. Assimilate. Okay, and the second chapter is mechanism of photosynthesis, especially light reaction. We already told you now light reaction and dark reaction. Photo light dependent reaction and light independent reaction. Okay, first light reaction we will discuss, and then dark reaction. That means assimilation of the carbons. Okay, carbon products, formation of the carbon products, carbohydrates. That are 3 cycles are there: C3, C4, and CAP. C3, C4, and CAP. Okay, that all are there. Where we are. Okay. C3, C4, and CAM. The first primary product is the 3 carbon that we call as C3 cycle. That also we also call as Kelvin cycle. Melvin Kelvin first identified, observed this reaction. For that, he got the Nobel Prize also. That is C3 cycle. The first, that is PGA, phosphoglyceric acid, 3 PGA, first product, 3 carbon product. That's why that is C3 cycle. And the C4 cycle, if the first product is 4 carbon product, oxaloacetic acid, it having the 4 carbons, oxaloacetic acid, OAA, that is C4 cycle. And the next one is CAM cycle, carboxylic acid metabolism. Carboxylic acid metabolism. That usually these happen in the xerophytic plant, succulent plants. Xerophytic and succulent plant, where the plants it close the stomata at the daytime. It absorbs the carbon dioxide at the night time and close the stomata in the daytime. Whatever absorb the carbon dioxide that will be utilized in the daytime for the photosynthesis. Okay. C3 cycle is a spatial differentiation, spatial separation. C4 cycle is the spatial separation, and CAM cycle is the time, time separation, daytime, nighttime. Okay, and another is the C2 cycle, photorespiration. C2 cycle. If instead of carbon dioxide, what happened? Instead of slide, could you able to see the slide? Photosynthesis. This slide is appearing.
Yes, sir.
Okay, okay, okay. Then photorespiration. If suppose in presence of the carbon dioxide, the RUBP, not RUBP, are you having the BP, are you having the sugar? Okay, BP, not BP sugar, RUBP,
Ribulose.
ribose, yes, 1,5 bisphosphate, RUBP. Hmm? Don't confuse, are you having the BP? Huh? RUBP. RUBP, ribulose 1,5 bisphosphate, it is the highest quantity of the protein enzyme which are present in the plant. Highest up to 40 percentage among all the protein, RUBP is present. That's why it is the highest amount of the protein or enzyme which present in the plant. So many competitive exam are here. Highest quantity of the enzyme or protein present in the plant is RUBP, PEPO, so many other enzymes, whatever it is. But which one is highest? RUBP.
RUB.
Okay. That RUBP, if it observes oxygen that goes toward the respiration as like a respiration instead of photosynthesis, respiration. It formed the two different that is 2 phosphoglyceric acid and 3 phosphoglyceric acid. Okay, that 2 phosphoglyceric acid will go to the mitochondria peroxisomes. Whatever the oxygen that absorbs the chloroplast RUBP, that will goes to the peroxisomes and mitochondria, and it releases the ammonia, and it calls for the amino acid synthesis, and it also releases the NADH plus for the energy sources, ITTs. Okay, that is the photorespiration. Okay, these reactions, these all we are going to discuss. Okay, you know the where the photosynthesis occurs, the photosynthetic site, where in the green parts of the plant. Where the photosynthesis occurs?
In thylakoid.
Uh, in the thylakoid, light reaction of the stoma
dark
Dark
reaction.
reaction.
Dark reaction.
Overall is chloroplast.
Fluorescence.
Overall, chloroplast. In the chloroplast, you see in the chloroplast. Not only in the leaf, photosynthesis will occur in all the green parts of the plant. All the green parts of the plant. Okay. If it occur only in the leaf, what about the algae? What about the bacteria? Some of the
But
photosynthetic bacteria. Photosynthesis occur in all the green parts of the plant. Not all the green parts of the animal, not all the green parts of the non-living beings. Only green, green part of the plant. Okay? You know, it is a phytochemical reaction, biochemical reaction. It is a master for all to exist the life. Okay? Here in the first topic is, what is that? First topic of our is,
Reading connection.
huh? You see, photosynthetic pigments and light
Light
harvesting.
harvest.
Okay? You see, for the light harvesting, what happens from where it is observing the light? Sunlight. Okay? In the sunlight, you know the sunlight is categorized into spectrum, light spectrum. You see the light spectrum. The light spectrum only 400 to 700, in between the 400 to 460 to 700 nanometer angstrom wavelength of the light only will be absorbed by the plant. Okay, how much it is?
390 to 760 nanometer.
Yes, 390 to 760
760.
nanometer wavelength light only will be absorbed. This is light spectrum of the photosynthesis. This much only. You see here, this much amount of the light, of the all light, of the all light, it is all the light spectrum of the sunlight. Other plants are observing only this much, only 2% of the light. Only 2% of the light are being observed by the plant. Only 2% of the light. Okay. You see the light spectrum. This is also very important. 390 to
760.
760. Okay. This is the light spectrum of this, especially which light is very,
Visible lights.
which light?
Visible lights.
These all we calling it as visible light.
Green. Green.
These all we call as VGR. Violet,
Red.
indigo, blue, green, yellow, orange, red. VGR. Visible light only. Visible light. Which light? Of the visible light, seven colors are there now. VGR, rainbow. Of that, which color the plant do the more photosynthesis?
Red and blue, sir.
Yes, blue and red. Okay, these two colors, blue and red color. These two colors majorly used for the photosynthesis. Okay, very good. Then we have to erase it. Okay. These are light spectrum. Okay, of the light spectrum, we are, you see the, what is the chlorophyll pigments are observing? Which light the chlorophyll spectrum, chlorophyll pigments are using? You see the chlorophyll A, B, carotenoids, which are observing how much wavelength? Which wavelength it is observing?
Between
Yeah, between 390 to 760. Of that,
760.
you see the chlorophyll B observing this in between these. Chlorophyll A observing in between these two nanometers. Whereas carotenoids and phycobilins observing in between these two. Extra pigments, accessory pigments, carotenoids
Okay.
and phycobilins,
Hmm.
accessory pigments. Okay, that also will be useful to harvest the sunlight. To harvest the sunlight. Okay, this is the spectrum. Okay, you see how whenever they absorb the sunlight, these are exciting and releasing the electrons. You know, whatever it is in the stable state, it don't give any energy, it don't participate in any action reaction. Okay, the stable condition. Whenever it get excited with the electrons or protons, you know, photon means
Okay. Light energy, sir.
light energy it having the protons and electrons. It having the protons and electrons.
Electron.
Okay. Here electrons are being used for the excitation. To excavate, to excite the molecule. Where which molecule here is chlorophyll. Chlorophyll. Okay. The chlorophyll will excite and it's releasing the heat and also releasing the photons. That proton and will be useful for the transportation. The photon transportation will lead to the formation of ATP and NADPH through the electron transport. Along with the electron, protons also will be transported. That transport will be very useful in the ATP formation, which ATP and NADPH only will be useful for the synthesis of carbohydrates. Okay, you know, oxygen when the oxygen is released in light
What?
reaction or dark reaction.
In reaction.
Yes, in the light reaction, oxygen will be released. Okay, the light reaction, not in the dark. Okay, the light reaction, especially photosystem one, photosystem two are there now.
Yes, sir.
In that photosystem one and two, with the one it releases the whatever the
Okay.
water that split into proton and oxygen, H plus and half molecule of O2, 2H plus half molecule of O2, oxygen. That especially in the PS1, you know, the cyclic and non-cyclic phosphorylations. You know now, cyclic and non-cyclic
Yes, sir. Only one photosystem is involving, that is the cyclic.
cyclic.
Two
Yes,
photosystems are involved, non-cyclic.
non-cyclic. Which one is the best one? Which one yields
Mm,
high energy?
non-cyclic, sir. Non.
Non-cyclic.
Okay.
Here it is participating two. PS1 and PS2 both
The
are
exit
participating.
of electrons also move.
Yes, yes. That's why it releasing the more. You know the significance of the photosynthesis. We all know very well. I already discussed. It is very useful for the oxygen production and useful for the food chain. You know the from the sunlight plants absorb and it form the carbohydrate. The carbohydrates and proteins will be eaten by the herbivores. Herbivores will be eaten by carnivores. Carnivores will be eaten by omnivores. Producers, primary consumer, secondary consumer, tertiary consumer. You know now. And also for the
Yes.
absorption, fixation of the carbon dioxide. If the more carbon dioxide is there in the atmosphere, what will be happen? Global warming, temperature, increasing of the temperature. Now you see in the European countries, temperature increase. They are uh, facing the
Heat,
so much of the problem
heat
with the
waves
heat.
are trapped.
Heat will be absorbed. If the more carbon dioxide, more heat will be absorbed. That cause for the global warming. So if we want to minimize the carbon dioxide level in the atmosphere, what we have to do? We have to plant more plants. We have to plant more plants so that more carbon will be absorbed. Carbon sequestration. Plants which absorb the more carbon dioxide. Okay, and also energy source for all the living organisms, and also main support for the biodiversity and ecosystem. If there is no food production, there is no biodiversity, no ecosystem. Okay, and where we are going to we have to study the pigments now. Yes, what are those? Pigments are chlorophyll, carotenoid, and phycobilin. We already studied it, this one. We already over now. Chlorophyll A, B.
Yes.
Huh?
Yes. Chlorophyll A,
Chlorophyll A.
B,
What is
C,
the
D.
formula of the chlorophyll A? C55, H72, O5, N4, Mg. Okay. And you see what is the difference between chlorophyll A and B? In the chlorophyll, where it is?
Carbon numbering చేస్తే
Yeah, you can see here. The chlorophyll pigment having the 4 pyrrole rings. It having the 4 carbons and 1 nitrogen. The 4 pyrrole rings attached 1, 2, 3, 4. Okay, and at the 3 sides, it having the phytol chain, 30 carbons phytol chain. Okay, here at the second carbon, CHO is there in the chlorophyll A, whereas in the chlorophyll B, CH2. Here the methoxy group is replaced by methyl group, CH2 group in the chlorophyll B. So what happened? One oxygen reduced. That's why C55, H72, O4 only, not O5. Okay, O4, N4, Mg. Okay, you can see here in the map that where it is, so that you can easily understand. Where? Where we are gone. Here you can see clearly. Okay, you see CH4 group is replaced by CH2. Okay, so that what happened? Instead of O5, O6. Oxygen, one oxygen is increased. Okay, then chlorophyll C, D, E, they don't have any phytol. Tail ఉండదు. Only 4 pyrrole rings ఉంటుంటాయి. Okay, okay.
Okay, sir.
Yeah. Here you can see the variation. These are the main variations of the ring. Chlorophyll A, B, C, D, E. C, D, E, there is no phytol. Okay, phytol, 30 carbon phytol chain is not there, absent. It present only in the A and B. Okay, A having the CH2, B having the CH4 methoxy group. Okay, at the second carbon, second pyrrole ring, third carbon position. Okay, and then you can see here in the D and E, it having the CH2, CH double bond, CH2, ethyl group. At the
Ethene.
first, ethylene. Ethene,
Ethene.
ethyl, ethylene. The 3 bond, that is ethylene. Okay, 2 bond, that is ethene. Only 1 bond, that is ethyne.
Ethene. Ethene.
Mm. Okay, that is a change. And then the carotenoids. Carotenoids are divided into 2. What are those? Carotene and xanthophyls. Carotene and xanthophyls. Orange color and yellow color. Carotene and xanthophyls. You see, carotene, they don't have the oxygen. Unoxygenated carbohydrate. Okay? But whereas xanthophyll having the oxygen. You see the formula. What it is? C40, H56, O2. The xanthophyll. Whereas in the carotene only C part is X56. Oxygen molecule is variation. Okay. And what the usage of these carotenoids? What is the main function? It absorbs and transfer the light to the chlorophyll A. Okay. And it protects
Hmm.
the photosynthetic system from the from the photooxidative damage. If the more light, chloroplast pigment will be damaged. That damage will be stopped by this accessory photosynthetic pigment. Main photosynthetic pigment is the chlorophyll. Accessory extra chlorophyll pigments are carotenoids and phycobilins. Okay, you know carotenoids, carrot, alpha carotene, beta carotene is present in beetroot. Okay, xanthophyll, yellow color. Okay, xanthophylls and carotenoid is sealed the chlorophyll pigment. And the phycobilins, you know the carotenoids are organic soluble, whereas phycobilins are water soluble pigments. It dissolve in the water. Okay, the open chain system, even in the carotenoids and xanthophylls. You have seen the structure now. You can see the carotenoids and xanthophylls. Leucine, carotene. Okay, phycobilins again two types: phycocyanin and phycoerythrin. Three and allophycocyanin. Usually these are present, mostly present in the algae. Okay, these also harvest the light and it supply light to the chlorophyll A. You know, chlorophyll A is present in all the photosynthetic organisms. Okay, chlorophyll A and B is present only in the chlorophyll. A and B majorly present in all the higher plants. Okay, A and B. Okay, and then we will go further. It is where we are. Light harvesting complexes. Where we are. You see, so many experiments are there that the experiments you already studied in earlier. No need to study. You see the visible spectrum. That spectrum is being used. That means light is compulsory needed for the photosynthesis. That is said by observed by Indian house. Okay, and you know the oxygen evaporates from the water molecule, not from the carbon dioxide. That as proved by Hill and Bendall reaction. You know the Hill reaction. Hill reaction. Okay, what the experiment he has done? He supply instead of the water H2S. You know sulfur bacteria, purple sulfur bacteria, non-purple sulfur bacteria that absorb the H2S. మురిగిన కోడిగుడ్ల వాసన వచ్చేదాన్ని సల్ఫర్ గా గంధంగా మార్చేస్తూ ఉంటుంటుంటాయి. Sulfide into sulfur. Okay, that is happening in the purple sulfur and non-purple sulfur bacteria. With that reaction is easily identified, whereas instead of H2O, H2S it supplies that releasing the sulfur instead of oxygen. That's why he identified, and also by using the O18 isotopes by using the isotope oxygen O18, actually O16 only, but increase the protons, two protons, so that O18 isotope. That O18 isotope, by using that, where the water molecule having the O18 isotope, that same O18 isotope, O18 is coming in the oxygen. That's why oxygen is releasing from the light reaction only from the water, not from the carbon dioxide. Water is oxidizing and carbon dioxide is reducing. That means oxidation, reduction reaction. Both the reactions are happening in the photosynthesis. Oxidation reaction and reduction reaction. Both are happening in the photosynthesis. Okay, you can see the light. What it is? Julius von Saxer, what he said? Plants that produce the starch. Okay, and the another experiments also there. So many experiments are there. Priestley, oxygen releasing. First, Joseph Priestley, oxygen releasing from the plant. He said, off-leaf experiments is there, and vigorant leaf experiments are there. So many are there like that. Okay, so many experiments are there like that. Now we are going to next step. That is pigments we have studied. Okay, next photosynthetic operators. What are those? PS1, PS2, PS1, PS2. What are there in the light reaction? Just now we have seen earlier. PS1, PS2. What you have seen? Are you there? You see, the PS1, PS2, the board, whatever is there, that is in the participating in the light reaction. You see, we divided into these are two, light reaction and dark reaction. We already studied the pigments. Absorption of the light spectrum also we have seen. Site of the photosynthesis also we have done. You see here, there is a light harvesting complex. PS1, PS2, and light harvesting complex. These three are the main phytochemical reactions, photosynthetic sites. Photosynthetic sites. The light harvesting complex, which absorb the light, especially the light harvesting complex PS1, photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2. You see the photosystem 1, it having the how many? How many? 200 chlorophyll and 50 carotenoids. And which one? P700. Which one it is? P700 is there. Usually in the photosystem 1, P700. That means it absorbs the light at the 700 nanometer. The photosystem it absorbs the light at the 700 nanometer. That is P700. That is present in the PS1. Whereas PS680, P680, that is present in the photosystem 2. Okay.
Okay.
These are the compounds. What are the compounds in the light harvesting complex? Chlorophyll, 200 above, and carotenoids, and photosynthetic site. P700 or P680. Okay. You see, and also cytochrome B6 and cytochrome F. How many cytochrome B6? How many? 563 cytochrome B6 and F complexes, and peridabsins also present. Okay. The PS1 it produced NADP. What it is?
Generations.
NADPH plus H. That means NADPH2. It produced the NADPH2. And also PS1 only, PS2. PS2 is having the same 200 chlorophyll pigment, and also how many? 50 carotenoids, and which one it is? Which side is? P680, already told you. Okay? And then plastoquinones and cytochrome B6 and F. Okay, here in the photosystem 2, water molecules split into oxygen and protons. Oxygen and protons. PS1 and 2 combinedly it cause for the production of ATP and NADPH. That is assimilation energy. What we calling it as assimilation energy, harvesting the light and it producing the ATP and NADPH. That light harvesting products by the light harvesting complex. Light harvesting complex, photosystem 1 and 2. What are there? Chlorophyll, that means pigment. Chlorophyll, carotenoids, and phycobilins. Pigments, chlorophyll, and photosystem 1 and 2. These all are the light harvesting complex product system. Okay, and we go for the mechanism of the light reaction. The light reaction, you see, the photolysis of water happen in the light reaction. That means in the light reaction, it produces the ATP and NADPH and splitting the water and releasing the oxygen. These three processes are happening in the light reaction. That process we call it as photophosphorylation. What process it is? Photophosphorylation. Here the phosphate ATP synthesis is happening. That's why phosphate, using the phosphate, it's synthesizing the ATP. That's why phosphorylation, which is happening in the light, under the light. That's why photophosphorylation. Oxidative phosphorylation, that is happen in the respiration. Photophosphorylation occur in the photosynthesis. Okay, don't confuse photophosphorylation, oxidative phosphorylation. Okay, you see here in the photophosphorylation, two cycles are there: cyclic and non-cyclic. Cyclic phosphorylation and non-cyclic. Cyclic means if it starts from the reaction center, that suppose P700, again it electron goes to the same. Again it goes to the same. That is what we call it as cyclic. It is cyclic. Where it started, there it ends. Okay. Where it started, there it ends. That's why we call it as that's why we calling it as cyclic. We are calling it as cyclic reaction. Cyclic photophosphorylation. ATP synthesis is occurring by transporting of the electron in circle. Where it started, there it. You see the PS1. Here it is started. It absorbed it, whatever the light harvesting complex, it excited and released the electrons. That electron is transported to electron acceptor. Which one is the electron acceptor? Here is the PS1. Pytin. Which one it is? The Pytin. The Pytin it absorbs the electron and it giving it to the FES. That means your ferrous sulfate complex. Okay, and then where it goes? Peridoxin. From the peridoxin to cytochrome B6 and F complex. Cytochrome B6 and F. The cytochrome B6 it is what it is? It absorbs the phosphate and releasing the ATP. ATP formation is taking place here. One ATP form the two electrons. Each electron one ATP will form. And then what the electron goes to phosphorus. The phosphorus is interlink carrier protein. It carries the proton electron from one to other. Here is the outer side of the thylakoid membrane. Light reactions will happen in the thylakoid membrane. Dark reactions will happen in the stroma. Okay. Grana stroma, the grana and chloroplast. Thylakoid chambers places we call it as grana. In the grana we have having the lumen, empty space. Empty space of the grana is called as what happened? Okay. Empty space of yeah. Okay. Empty adapter. Here it is again entering into the PS cyclic. Okay. Here it is the only ATP, not NADPH. Only ATP is forming, electron coming again back. Okay. Okay. Here reduction and oxidation reaction both are happening. That's why redox, not redoxide. Okay. Redox. Oxygen reduction. Oxygen oxidation and reduction reaction. Okay. Then whereas the non-cyclic what will happen? All of you, are you there? Are you there? Lokeshwari, Lakshmi Prasanna, Lavanya.
Yes, sir.
Okay, okay. You please respond. Otherwise, whether you are there or not, I confuse. Okay, where we are? Here. Okay. Then the non-cyclic. Here in the non-cyclic, the both PS1 and PS2. Photosystem 1, photosystem 2, both are being in participate. Here, whatever the first electron, the light harvesting complex, that start from the PS2, that start from the PS2, and here with that light energy, it is fitting the with that light energy, it is fitting the water molecule. It is fitting the water molecule. Here it is fitting the water molecule. You see, H2O. Photolysis is happening here. H2O is fitting into the H plus oxygen. This is the basic fundamental best reaction in the photosynthesis. With this reaction only, we could able to get the oxygen. Oxygen is releasing in cyclic or non-cyclic phosphorylation. Non-cyclic phosphorylation, oxygen is releasing. Where it is releasing in the PS2 system 2. Okay. That is light harvesting complex. Light harvesting complex. Whenever light is absorbed, it is releasing the electron. With that electron is being activated. That activated light harvesting complex, splitting the water molecule into H2H plus and oxygen. Okay, and then electrons are being absorbed. The primary electron acceptor, that is here from there it absorbing the cytochrome B6 complex. Here same as like the cyclic reaction, cytochrome B6 it all is ATP, same as like cyclic. But ATP formation, the cytochrome complex, the electrons are going to where it is going. The plastocyanin, this plastocyanin supplying the electron into the sending electrons into the PS1. Which one? PS1, 700 nanometer. This is a 680 nanometer. Then the PS1 again it is going the electrons to the primary electron acceptor theopitin. From there again it's going to where it is going. This electrons going to peridoxin. Where it is? Peridoxin. Here electrons absorbing by peridoxin. This peridoxin also participate, also absorb protons. Further protons here, whatever the protons which are released, that protons will be absorbed and it form the NADH2. It form the NADH2. Okay, so this NADH2 is coming in the PS1 at the end of the light reaction in the non-cyclic. Non-cyclic reaction also called as Hutch-Slag cycle. Hutch and Slag. Hutch and Slag first observe this cycle. That's why we call it as Hutch-Slag cycle. Hutch-Slag cycle. Okay. Here in the non-cyclic, ATP formation, oxygen, releasing of the oxygen, and NADH2 formation. These three reactions happening in the non-cyclic. Okay. Whereas in the cyclic only ATP formation will take place. In the cyclic only one ATP. Whereas in the non-cyclic ATP, NADPH, and oxygen. These three. That's why PH2 is highly productable reaction. And that means non-cyclic is a highly productable reaction. Then the cyclic. Okay. Then the cyclic reaction. There is a cyclic reaction and non-cyclic reaction. You know very well. Essay question will come. Non-cyclic. Majorly essay question asks the non-cyclic. Hutch-Slag cycle. Okay. And then we will go for the dark reaction. Hello. Dark reaction. Dark reaction means it is not happening in the dark. This is not happen in the night. This also happen in the daytime only, but independent of light. No use of the light. Already light reaction which are utilized, light reaction by using the light, it produces the ATP and NADPH. Hello. ATP and NADPH, accumulation energy. That accumulation energy will be utilized here in the dark reaction. So you have no need of the light. Whatever the light energy is converted into chemical energy, biological energy. That chemical energy or biological energy. Hello? Ah, I am in the class. Why you are taking the class again? Call again and again. I will call you. I will call you, please. Okay. You can see here the dark reaction. We call it as light independent reaction. The light independent reaction, you can see what it is. How many steps are there? You see the first is carbon fixation, reduction, and are you there? Carbohydrate synthesis and regeneration of RUBP. Okay, the main four reactions which are there. You see the dark reactions again having the three types. Again having the three types. What are those three types? What are those three types? C3, C4, and
Cam.
CAM. C3, C4, and CAM. These are the three types. Okay. There we are. Okay. These are the three types: C3, C4, and CAM. There we are gone. Three cycles at a place. I kept at a place. No, three cycles. Yes, here you can see C3, C4, and CAM. Three different cycles are there in the dark reaction. Okay, C3, here it is first carbon production, stable production is 3 carbon products, that is 3 PGA. That's why we call as C3 plant. Here in the C4 plant, first stable product is oxaloacetic acid. It is a 4 carbon product. That's why we call as C4 cycle. Here it is CAM plant, carboxylic acid metabolism. It is happening in the xerophytic plant, desert plant, halophytic plant. Okay, there daytime it close the stomata and nighttime it open the stomata. Nighttime only it absorb the carbon dioxide and it assimilates it as a malic acid and then the succinic acid or malic acid, and then that will be participate in the decarboxylation so that carbon dioxide is released. That carbon dioxide will be participated in the C3 cycle again. Okay. In the light reaction, major utilization is the water. In the dark reaction, major utilization is the carbon dioxide. Okay. Here carbon dioxide reduction is taking place. There water oxidation and reduction will take place in the light reaction. Okay. These are the three different cycles. We will go each and one. Okay, first one is the C3 cycle. We also call it as Kelvin cycle. We also call as Blackman reaction. We also call as carbon reduction cycle. Carbon reduction cycle. Okay, these are the so many names are there. You see in this reaction what is the main steps are there. The first product is PGA, 3 carbon product. phosphoglyceric acid. That's why we call it as C3 cycle. And here, four steps are there. First is carbon dioxide fixation. Second one is carbon reduction. Third one is carbohydrate synthesis. Fourth one is regeneration of RUBP. Regeneration of RUBP. This is the total overall reaction. Overall reaction of the C3 cycle. The first one is absorption of the carbon dioxide. First step is fixation. Carbon dioxide fixation. You see, you see here what happened. What is happening? What I told you, the 40% of the protein which are present in the plants of plant is RUBP. RUBP, ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate. This ribulose 1,5 bisphosphate is absorbing the carbon dioxide. It's absorbing the carbon dioxide. Okay, carboxylation reaction. Carboxylation reaction. And it is from the 2-3-PDA, two molecules of 3-phosphoglyceric acid. Phosphate at the third carbon. That's why 3-phosphoglyceric acid. Okay, here is having the 1, 2, 3 carbons. That's why we find as C3 cycle. Okay, carbon in place. The 5 carbons in the ribulose phosphate and 1 carbon. 5 plus 6 carbons. Here it is formed 2, 3 carbon products. 2, 3 carbon products. That is 2, 3-PGA, phosphoglyceraldehyde. Okay, and that absorb the ATP. Okay, and it formed the 1,3-phosphoglyceric acid at the first carbon and the third carbon. Both the sides, 1 and 3 phosphate. Already at the third carbon phosphate is there, and also first carbon also this phosphate be added from the ATP. Okay, that's why this we call it as 1,3-phosphoglyceric acid. This reaction we calling it as reduction reaction. Okay, phosphorylation, one phosphate extra added, and this 1,3-phosphoglyceric acid again is in the ATPase2. Where from it generated? NADH and ATP are generated in the light reaction. Accumulated energy. That light reaction accumulated energy is being used in a dark reaction. Okay, the 1,3-phosphoglyceric acid is absorbed protons from the NADPH plus H and it is transforming into glyceraldehyde 3-phosphate. Which one it is? Glyceraldehyde 3-phosphate. Aldehyde group. It changes into aldehyde group. Okay. The glyceraldehyde 3-phosphate, it is phosphating in isomerization reaction. Fourth reaction is isomerization reaction. In this isomerization reaction, glyceraldehyde 3-phosphate will be changed into dihydroxyl phosphate. One glyceraldehyde 3-phosphate will change into DHA, aldose group, ketose group. PGA is the aldose group, and dihydroxyl phosphate ketose group. You know the keto group aldehyde group. If it is a slow group, like the carbon is ketone group. And aldose should become ketone. Okay, these are smallest sugar compound. Smallest sugar component are the what is it? PGA. Smallest sugar compound. Three carbon product. There are here three the smallest sugar component. Okay? And here, what happening here is these two glycerol, one glycerol 3 phosphate, one dihydroxystone phosphate is coming as a fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. Are you there, Abno? Are you listening? Are you listening? Are you there?
Yes, sir.
Okay, okay. Then fructose 1,6 bisphosphate again it releasing the one phosphate from the first carbon. Again it forming the fructose 6 phosphate. Fructose phosphorylation reaction fructose 6 phosphate. Okay, here P substituted with the hydrogen proton. Okay, this fructose 6 phosphate again with the one glyceraldehyde 3 phosphate is forming the what it is forming ethyl 4 phosphate and xylose 5 phosphate. What is ethyl phosphate? Second one is xylose 5 phosphate. One bicarbon, one four carbon, 9. Here is 6 carbon, this is the carbon. 6 plus 3, 9. Here into 4 plus 5. Erythrophosphate phosphate and xylophosphate. Okay? And then the erythrophosphate, dihydroxystone phosphate is coming the 7 carbon product. Again, this phosphate release the one phosphate and it form the pseudo-H 17 bisphosphate into pseudo-H 7 phosphate. One phosphate is released here, inorganic phosphate. Okay, then you see the pseudo-H 7 phosphate with another PGA, phosphoglyceraldehyde 3 phosphate. By mixing this, it is forming the xylulose 5 phosphate and ribulose 5 phosphate.
Ribulose.
Xylulose, one is the xylulose, second one is the ribulose.
Yes.
2 5 carbons, 5 plus 5, 10. Here the 7 plus 3, 10.
10.
7 plus 3, 10 here. Soda plus 7 phosphate plus 3 glycerol 3 phosphate. Here it is split into two half equal parts. One is xylulose 5 phosphate, one is ribulose 5 phosphate. Okay. Here again this ribulose 5 phosphate is what it is? Isomerization reaction. It is changing into ribose 5 phosphate. Ribose
Right.
5 phosphate. Ribose 5 phosphate is become changing into ribose 5 phosphate. And also here you see xylulose 5 phosphate. This xylulose 5 phosphate also will be changing it as a ribulose 5 phosphate. So from here ribose 5 phosphate and xylulose 5 phosphate. The two ribulose 5 phosphate. Again what happened here? What do you observe? RUBP formation. Again RUBP form.
Yes.
Again RUBP form. What is the byproduct here? Here they use carbon dioxide and ATP and ADPH. Yes? These carbon dioxide and ATP and ADPH are being used for the production of glucose. For the production of glucose, here you see the fructose. This fructose 6 phosphate form now. One fructose 6 phosphate will be used converted into glucose. Remaining one fructose 6 phosphate is being participated in the cyclic reaction. Okay, this fructose 6 phosphate with the xylar 5 phosphate, it formed the pseudo, what did it form? Pseudo plus 7 phosphate. But one fructose 6 phosphate is changed into glucose 6 phosphate. That glucose is the byproduct here. This is the byproduct. That means so here it absorbs 6 carbons. Here it is forming the 6 carbon product, glucose. Okay. 6 carbon dioxides absorbed, 6 carbon glucose products is releasing. No usage from the RUBP. RUBP molecules are not being used in the glucose production. RUBP again, it reformation is taking place. For the 6 carbon dioxide, 6 glucose molecules, C6H12O6 are formed. This is the C3 cycle. First product is the 3 carbon product. That's why we call it as C3 cycle. Okay. The Kelvin cycle, very, very important essay question. It having the 13 steps. You should remember this all the steps. Okay. And then we will go for the C4 cycle. The first byproduct is the oxaloacetic acid, 4 carbon products. First byproduct is the 4 carbon products. That's why we call it as C4 cycle. Okay, here you can see here clearly. You can see here. This you can say, take the screenshot also. Now clearly you can see how from where it is happening. Here it is the byproduct. Okay. Here are the byproduct glucose. These all are again forming the ribose phosphate. Here the first reaction is carbon fixation. Second reaction is reduction. What it is? Carboxylation. And third one is reduction reaction. Reduction is taking place here. NADP into NADP into NADP. Hydrogen molecules losing. That's why reduction reaction. And again regeneration of RUBP. 4 reactions. First one is carbon fixation. Second one is reduction reaction. Third one is carbon carbohydrate formation. Fourth one is regeneration of RUBP. The 4 reactions are there. The 4 reactions. Okay. You know the steeper plant. We also call steeper plant is from the spectrum diet and the bundle seed Frange anatomy. Frange means garland. The Japan called Frange means garland. There you can see the itself is covered the cells cover the which one is covering vascular to prevent from the wastage of the water. That not is a special character for the C4 plant. All the more parts are C4 plants. The C4 plants are more carbon absorbed plant than the C plant. Whereas in the C3 plant, carbon fixation is taken place with the RUBP. The C4 carbon fixation is taken by the phosphoenol pyruvic acid. Here is the C4 side RUBP utilization. That's why there is no wastage. of the products. There is no wastage of the products. All product will be converted into glucose formation. That's why more highly productive cycle is C4 cycle. Okay. There is a lot of variation between the C3 and C4 cycle. The C4 the C3 plant first product is 3GA. The C4 plant first product is the C3 first fixation molecule dipose one. The C4 cycle first accumulation fixation is the pepper. Okay, like that so many are there. Here you see the C4 cycle. Okay, here C4 cycle is the spatial separation cycle. Spatially separate. Spatially separate. Means some part of the reaction occur in one place, remaining part of the reaction occur in another place of the greenery place. You see in the leaf, mesophyll cells and bundle seed cells. Okay, mesophyll means these are the remaining mesophyll cells. Between the upper and lower end the mesophyll cells. In the mesophyll, the first reaction will happen, and in the bundle cells, second reaction will be spatial variation is there. The mesophyll cells absorb the carbon dioxide, and the bundle cells it occur the C3 cycle, Krebs cycle. Krebs cycle occur in the bundle seed cell, and as the association of the carbon will be the mesophyll cell. Okay, here see the phenol pyruvic acid. Phenol phosphoenol pyruvic acid is the primary carbon dioxide observer. It absorbs, you see here the phosphoenolic acid, it releases the ATP and is forming the phosphoenol pyruvic acid. It absorbs the atmospheric carbon dioxide, you see the carbon dioxide, and it's changing into bicarbonate, HCO3, bicarbonate products. That bicarbonate product absorb PEP, PEP plus